today I want to share with you is a research area inspired by Eric Olson's pioneer work and in the area of postnatal cardiac maturation. And some of you may already know that, I came to the United States from China as an undergraduate, and I went to Baylor College of Medicine to start my PhD. But to me, I didn't realize at that time is that in my thesis committee, I need an outside member. And I look around the catalog of faculties who knows genetics in Houston, and I found this young professor called Eric Olson. And he was gracious enough to actually join my thesis committee and really being such a generous mentor and everything, gave me so many insights and guidance throughout my career. And that's why I'm triply honored to actually deliver this lecture after his seminal work and the presentation just last episode. So again, I want to thank everyone for the great support. And I, of course, can't present anything without really the incredible members of my team. And I think I need to hide this one. Yes, so you can see. Both from during my transition, the UCLA team, as well as the current one at Duke and US in Singapore. But I really like to highlight a few who actually really at the core of our of my presentation today, which is a relatively new area in my lab that actually include Dr. Gao, who is now assistant professor at the University of Cincinnati, Dr. Marlene Torma, who is associate professor at the UCLA Pediatric Department. Christopher Rao is assistant professor at UNC. Tomo is now assistant professor at the VA of UCLA, and JJ is a senior fellow at UCLA Department of Medicine. So I really wanted to make a tribute to their incredible talent and efforts and dedication, who really benefit and enriched my knowledge and truly inspired me every day to continue many of the lines of work that we're doing right now. So, of course, we share the same interest in many of us in this audience about heart, which is very important organ, vital, and even in the midst of medieval time and ancient times, we put the heart in a very important position. And of course, now we know a lot better that heart function is so important, is because it provides really the source of energy to circulate blood in our body, and it has many incredible properties. And the more I study it, the more I really become very, I would say, very respect to this organ. And our current interest is really to understand the complexity of the heart function dysfunction remodeling, especially in the context of metabolic disorders. And in that regard, continue work on the branch amino acid in our focus, and hopefully have another chance to report to Henry, rekindle our interest in this area. This is certainly still a very active research area in our lab. But what I hope to accomplish today is to highlight a kind of a new line of research in our lab, and that really as part of our overall interest to discover and characterize novel molecular players in the heart, and then put it in the context of cardiac disease and development, and hopefully to discover new intervention for human diseases. And that, again, interest about today's research topic indeed came from the pioneer work from Eric Olson and many others in the field, that they have defined the incredible, interesting process of cardiac development from just crescent, a line of cells, all the way to a very complex tube into chamber until after birth. And when we look at the heart, this is a very beautiful review from Dan Kelly, very recently published in JMCC 2024, showing in the circle of life in cardiac maturation and the failure, where the fetal and the postnatal young hearts indeed adapted to many common features as we observe in the failing heart. Still, the underlying mechanism or the underlying reason, what I call it, sort of the physiological significance, still puzzling to us. But nevertheless, if you look at many features in terms of metabolism, contractility, cell cycle regulation, and you can see there's a dramatic change during the fetal to adult heart transition. And subsequently, many of the features actually revert back to the heart failure. So at the original beginning, our very naive thinking is, we understand the heart failure go to the normal heart into a diseased heart, adapt to this so-called fetal-like features. And so if we understand from the fetal to adult maturation, maybe it can shed some light into the reverse process. Hopefully by the end of the presentation, we can share with you that this kind of connection really actually works in many layers, not just gene regulation, but also in signaling as well. And so when we think about, again, the cardiac maturation, if you look at the whole process, in the early stage of cardiac termination, it was very much driven by a transcriptional sort of signature. It's almost clearly defined by transcription regulation. But once you go from the early cardiomyocytes, sort of fetal cardiomyocytes or determined fully differentiated cardiomyocyte into adult myocytes, and this definition become all functional. We look at the morphology, look at electric physiology and metabolic activities, but the underlying mechanism remain poorly understood. And I just wanted to emphasize that there are two aspects of this cardiac maturation during this very short period of time. One is at the individual cell level, as we indicated earlier, from the morphology to different aspect, almost every aspect of cellular physiology. The other aspect is really within the heart, fetal heart to adult hearts. There is a dramatic change in terms of the dimorphic trajectory of growth in between the two chambers, the left and right. And of course, this makes perfect sense in terms of the physiology. Let me just bring the laser. This of course play a very important role in adaptation to the separation of the circulation in peripheral. One is, of course, for the much higher afterload of the peripheral circulation. The other one, of course, this is the right ventricle for much lower afterload in the pulmonary circulation. And so this is adaptive, or it is actually a proactive reprogramming between the left and the right, remain also unclear. And so this, at the beginning, when Tomo actually joined the lab, and he looked at very carefully in this period of time, looking at the several features that potentially can contribute to this left and right, which are the focus on my first part of the study report here. And what he observed is that by day 3 to 7, there's a clear separation between the left and right in terms of hypertrophy, proliferation, and even at the P1 level, there's a transient difference of apoptosis. So there are more cell deaths, lower proliferation, and a higher and lower hypertrophy, and of course that contribute to what we believe is the differential outcome in the left versus right. So this is what we observed from the morphological level into now cellular sort of a mechanism, but what is the underlying molecular mechanism is still unknown. And Dr. Marlene Torma, who actually initiated this line of study when she was a fellow in our lab, performed very comprehensive transcriptome studies and highlight the difference between the P0, between the left and the right, which is pink versus blue, along the developmental process during this period of time. And what strikes all of us at that time when we look at the data is that this left and right difference are significant, but only established at the whole transcriptional level right after birth. And remember, the left and the right actually are supposed to come from different origin of the developmental developing embryo. And so they actually, in theory, coming from a different sort of a genetic origin or a set of genetic information. Yet at the P0, it's actually very, very hard to distinguish them from the whole transcriptome level. And yet the separation actually become quite obvious. So that become the motivation for us to really focus on this postnatal period in order to decipher what could be the possible mechanism involved in the left and right differences. We know that differential, the MAP kinase actually involving many of those processes, including the growth as well as proliferation, as well as apoptosis. So at the beginning, we look at the MAP kinase pathway simply because we were at that time quite interested in this line of cell signaling. And again, when Tomo looking at the MAP kinase, ERK and the JNK, and there are no differences between the left and right, despite the difference in terms of their proliferative and hypertrophy status. However, when we look at the P38, which is the third branch of MAP kinase, we saw a dramatic difference between the left and the right heart, left and right, across this very critical period of time. Actually, after in adult hearts or in the neonatal hearts, and we actually don't see much of a difference anymore. So this critical period of time, they actually do present a very differential activity. And here is just some of the quantification. And so with that, it's as I indicated earlier, we found that this P38 originally being studied very extensively in the adult hearts. We know that this is part of a stress pathway in response to many of the mostly pathological related to stresses, oxidative and pressure overload and so forth. And we also know that this P38 MAP kinase, when it's activated, actually exerts almost universally bad things, including loss of contractility, promoting pathological hypertrophy and remodeling and loss of mitochondrial activity. So there are many, many aspects that actually P38 contribute to pathology of the heart. And indeed, the P38 inhibition has been clinically tested as a potential therapy. And so how the P38 potentially can contribute to this chamber-specific postnatal development? Tomo asked that question by making the P38 alpha and beta, the two major isoforms expressed in the heart, knockout specifically in the cardiomyocytes using a Krelux-P mediated mechanism. Actually, you can see that this cardiac specific knockout were quite robust, and we lost almost all the detectable PCDA expression. And immediately we observed very interesting differences in the surviving embryos, where before the birth, this breeding is supposed to give us 50-50 Cre positive and negative feature distribution, which we essentially observed. But once we go beyond the birth, there's a significant sort of a skewing towards the loss of double knockout mice. And so Tomo observed the survival embryos, the fetus, sorry, the newborns. And it was very obvious from the morphology, there's something wrong. And when you look at the histology, and clearly what we observe is now is the right ventricle is almost as thick as the left ventricle. And just compare with the controls. This is actually the day 0, and here is actually day 1 month old. And as you can see, the difference that we observe in the normal heart actually disappeared. And so Tomo actually measured, this is just by the measurements of the weight, and you can clearly see that this effect of change quite specifically impact on the left ventricle, right ventricle, but not left ventricle, right? So increased the RV weight without much of impact on the LV weight. And if when we look at the hypertrophy, apoptosis, and proliferation, everything he measured actually turned out to be impacted by the PCDA. Essentially, when a PCDA is knockout, I just skip some of the data, is that the hypertrophy is now equal, the proliferation is now equal, and the apoptosis actually disappeared. So now the left ventricle and the right ventricle become almost equal in size in terms of the thickness, at least. And so when he look at the gene expression, and not surprised that we see predominantly the genes activated in the knockout RV are related to cell cycle regulation. They are pretty much very dominant, almost across the entire geo priority scores. And that turned out to be something that we actually observed earlier in collaboration with Mark Keating, and they actually been screening for different molecules and pathways that can promote cardiomyocytes years before. And when they hit the PCDA, they indeed found that PCDA inhibition can promote the cell cycle in neonatal cardiomyocytes as well as adult cardiomyocytes. But we don't know at that time what is the potential mechanism. And so Tomo actually dive more into those genes that are actually affected by PCDA in the right ventricle. And what he observed from those genes that are represented by cell cycle regulation, if you look at upstream transcription factors, they also are populated by the common transcription factors that we know are the master regulators of cell cycle regulation. However, he also observed another player, which is kind of interesting and unexpected, that is the XBP1. And XBP1 is in an entirely different a stress regulatory pathway that is the ER stress, and it's the downstream of IRE1 and mediating this XBP1 splicing and non-canonical splicing and generating this functional mRNA, and then the XBP1 protein can be produced as a transcription factor to produce the gene expression. And although classically implicated in ER stress to promote sort of ER stress related genes, it is also being demonstrated to activate cell cycle. So we thought maybe that is also part of the downstream contributor to PCA mediated cell cycle cardiomyocyte proliferation. And so at least in the double knockout, what we observe is in the RV, now the IRE1 expression actually remains high, and the splicing XBP1, which is a readout of the IRE1 activity, also significantly higher than the counterpart, the genetic control. And so that gives us the potential sort of a hypothesis that maybe XBP1 is the downstream of PCDA that regulates the cell cycle regulation. And so in vitro, Tomo actually proved that indeed, if you inhibit the PCDA, as we demonstrated before, it produced higher level of cell cycle entry. And then if you overexpress IAE1, which is upstream of XBP1, you can achieve similar effects in terms of promoting the cell cycle in cardiomyocytes. But if you knock down the XBP1 in the presence of PCDA inhibition, and then what you see is that this PCDA inhibition induced cell cycle induction is diminished. So that proves this sort of upstream-downstream effects relationship. And to make it a further demonstration in vivo, we take advantage of this RE1 transgenic mice established in the lab, and to induce this expression in neonatal cardiomyocytes by expressing with the NCAS 2.5 crete. And as a result of this activation, what we observe is indeed the right ventricle start to actually getting much bigger, and in both chambers that showing this sort of enlarged phenotype. And so that again supports the concept that XBP1 expression or activation is sufficient to promote cardiomyocyte proliferation. And this observation actually can be potentially generalizable, because in a separate study across all multiple cell system in a mouse organ by single cell sort of atlas profiling, what they observe is that XBP1 is one of the common transcription factors related to cell fate determination, as well as cell, especially in the postnatal period. So we were thinking that this XBP1 observation may be not unique in the cardiomyocytes, but also involving other aspects of cellular development. And so this gives us again this the pause that you can see that the PCDA dependent signaling pathway in adult hearts is involved in the hypertrophic cardiomyopathy and many of the features of cardiomyopathies. However, when we look into the developmental phase and realize that PCDA is actually quite important, it is being utilized or activated as a genetic program to shape the heart between the left and the right postnatal growth and the proliferation. And so that's the first kind of a story that we learned, that the many of the things that we learn in the adult hearts actually has the developmental origin, particularly around postnatal period. And so let me switch gear to another aspect of cardiac maturation in the postnatal period. That is, if you look at individual cells, they are in the immature state during the fetal development versus the adult cardiomyocytes. And it's very clear that there are dramatic differences in morphology, cell physiology, metabolism, gene expression, you name it. And then in fact, we have more or less cracked the code of turn the stem cell, almost any source, into cardiomyocytes by a cocktail of manipulation, mostly focusing on transcription factors. But if you, no matter what's the source of the stem cells, those cardiomyocytes derived from those stem cells are universally immature in terms of their phenotype. And we still have a lot of challenge in the field to actually promote this cardiomyocyte maturation further, so that, which we know that is very important for disease modeling or potentially cell therapy. And so in the field, when we survey the literature, indeed, most of the methodology to promote the maturation is more or less a sort of what I view as the anecdotal external stress mediated manipulation, whether it's metabolic, whether it's mechanical, whether it's electric, and whether it's a prolonged incubation. All those actually kind of thinking using the external environment that mimic the maturation in process in order to promote this maturation process at the cellular level. But if you look at from the intrinsic mechanism perspective, and we know actually quite limited. We don't have much of a major insight except the very few studies that in the recent past. And so we, at that time, in a very naive sense, tried to use gene expression to look for the molecular signature associated with the maturation process during this period of time. And not surprising that we see much of the genes affecting the metabolism, including the branch amino acid, but also the other, especially the lipid metabolism induction and the glucose metabolism reduction. But in the negative regulated genes, of course, cell cycle, which is the hallmark of maturation process, that they exit the cell cycle. But we also interestingly see a major signature that involving RNA processing. But when we look at the RNA processing, especially splicing genes, which is highlighted in those markers, and you can see that vast majority of the RNA splicing factors are downregulated, except one, that is RBFOX1, quite significantly induced during the maturation period within this short window of maturation. And so that reminds us the early work about RBFOX1 by Chen Gao when she was starting that as a graduate student. And so what is the RNA splicing? And we know that RNA has this, mRNA at least has this interesting sort of a metabolism life cycle where it's produced and processed and spliced to generate the different isoforms, and then subject to either mRNA translation or degradation. And in this process, the mRNA splicing is carried out by this very conserved RNA splicing machinery, incredibly efficient, and they are coupled with transcription process and very efficiently splice out the intron and generate the mature mRNAs. But in this process, when interacting with regulatory factors, and they can generate different splicing product by skipping, by using different alternative 5' or 3' sites, so that you can generate a variety of we call the splicing variants. And as a result, when you look at any genes in our genome, the annotated gene product usually is far more than annotated gene product in terms of mRNA is far more than the gene number because of the splicing variants contribution to that complexity. And so Wang Chen actually looking at the splicing regulators in heart failure, and number one, she actually observed that many genes get spliced differentially, and this differential splicing is also mimic the developmental stage of maturation and also revert back in the heart failure state. So you can see that those two categories of splicing events. And when she looked at those splicing events associated with potential RNA binding proteins, we can see some of them actually are enriched. That means that those are potential sort of a transacting splicing factors, just like we see gene expression and try to guess what kind of transcription factors involved. But when she scanned many of them, and only one of them really panned out very consistently. That is RBFOX1. So it's support. It's kind of very interesting when we look at developmental maturation phase. RBFOX1 is also bucking the trend with other splicing factors to be induced, but in the diseased heart, it is also one of the unique ones that actually got downregulated specifically and remarkably, actually. And so Chen actually went ahead using various of genetic manipulation, demonstrated that if you genetically block RBFOX1 expression, you can promote the onset of hypertrophy and heart failure. And she also did reverse by restore the RBFOX1 expression, can ameliorate the onset of heart failure. So how the RBFOX1 expression correlate with the postnatal maturation then? So we validate the RNA-seq observation by RT-PCR and then showing that this is indeed induced dramatically from almost undetectable to a very high level. And if you look at the protein, that's exactly the same. And just to remind you that this induction was very early, dramatic, and significant. And so we used another data sets from RNA, single-cell RNA-seq data across the same time frame, provided by Professor Wang's lab from Beijing. And the one JD actually looking at this published the data sets of just the chondromyocytes along with the developmental stage from P1 to P14. And the RBFOX1 expression is almost exclusively located in this very small portion of the chondromyocytes, the CN5, which has the most mature gene expression profile compared to other subclusters, including the myosin heavy chain expression and the loss of cell cycle genes and so forth. And so that leads us to this hypothesis, maybe RBFOX1 is correlated and contribute to chondromyocyte maturation in this postnatal phase. And working with Josh Lee, a graduate student, and JJ and Josh, generate a series of observations where RBFOX1 expression in neonomyocytes can increase the cell size, but also increase the maturation associated with genes in terms of sarcomere protein, the calcium regulatory genes, and also promote the sarcomere organization and promote even the bionuclearization. And in addition, they look at the function and by contractile profile as well as calcium signaling. And the RBFOX1 expression can promote the contractile function consistency and as well as the calcium cycle in terms of timing. So you can look at the measurement of the time to peak as well as tau. They're very, very consistent throughout the experiment. And you can also look at action potential, and knowing that in the rodent cells, maturation of the action cells associated with the shortening of the action potential, you can see that RBFOX1 also significantly reduced action potential duration. This is in collaboration with Reza Hardy's lab. And this is the neonatal cells from rat, and when we shift using human iPSC-derived chondromyocytes, we generally observe very similar outcome in terms of sarcomere organization, gene expression, as well as improvement of contractility and the calcium cycle robustness. And so we can put the cells onto this instrument where we can measure the force of contraction as well as essentially the fraction shortening and on the matrix. And so against this resistance, and the chondromyocytes expressing RBFOX1 is more robust in terms of contractile function. So we believe that this can translate to a better more mature cells, allow for either drug discovery screening or mechanism studies. We also work with our collaborator from A-Star to utilize a interesting organoid, 3D organoid developed in Bone Sense Lab, where RB-FOXP1 expression with a lentivirus can also significantly increase the expression of marker genes associated with maturation, but also improve the sarcomere organization if you measure by the Z-line fraction as well as skewedness. So they are more organized and more mature, closer to the mature cells in this organoid. And so what's the underlying mechanism? We're still in the process of actually exploring that. In terms of downstream, Chen actually indicated in the neonatal, in the adult heart, RB-FOXP1 is affecting almost a quarter or more of the heart failure associated with RNA splicing events. Indeed, that if you knock down this RNA splicing event, it will be aggravated, but if you overexpress, you can almost completely reverse those heart failure associated with RNA splicing outcome. And in the interest of time, I would not deliberate more on the downstream mechanism, except that Chen actually identified the downstream, one of the downstream, at least the target is the MEF2, different isoforms, different family members of MEF2 that are the subject of RB-FOXP1 dependent RNA splicing. And so in this new paradigm of both external forces or chemicals that promote cardiomyocyte maturation, and now we're proposing that there's an intrinsic sort of transcriptome modulator mediated by RB-FOXP1 that potentially contribute to splicing dependent maturation regulation in heart. You probably have noticed that RB-FOXP1 expression is dramatically induced in the postnatal period. And this is at a protein level, and I show you also in the mRNA level. And so that raised the question of how this is being regulated, because knowing that this postnatal maturation gene expression is really a mix, it's a really a miss still to the field, and how this postnatal transcriptome gene activation can be accomplished. So we thought that this RB-FOXP1 may actually serve as an interesting probe for us to interrogate and identify this question. And JJ obtained the results from Bill Poo of P300 ChIP-seq data in the fetal to postnatal mouse heart. And it was very interesting to see that in the upstream, almost 10 kb away from the promoter of the RB-FOXP1, cardiac RB-FOXP1 promoter. And we observed this P300 peak only show up in the postnatal period and not detectable in the fetal or embryonic stage. And so this peak of P300 is also associated with other markers of epigenetic modifiers in terms of histone modification signature, supporting that this is perhaps a real chromosome open area and almost like a super enhancer. And it's tissue specific. We know that RB-FOXP1 is targeted to be expressed in two major tissues. One is in muscle, the other one is in brain. And so when we look at the promoter for the brain, there's no such signature of enhancer-like feature compared to the heart. So it's tissue specific. And it's also highly conserved. If we look at the mammals and this conservation sequence actually overlap quite well with this particular enhancer area. And it's also expressed eRNA from this particular region, and this being tested from C2C12 differentiation in vitro, again suggesting that RB-FOXP1 expression is associated with this opening of the enhancer or super enhancer activities. As to demonstrate that conclusively, JJ actually cloned this enhancer and narrowed down to almost now just a couple hundred base pairs. And he made multiple copies, and he can see that in vitro using C2C12 mild blast differentiating tumor tube sort of in vitro model, that this construct, only when the enhancer with its own promoter will drive the expression in the maturation stage. And when he put into the mouse, making transgenic mice, we were so excited that indeed this enhancer sequence allow the marker gene, this is TD tomato, express only specifically in the postnatal period, and also in muscle as well, only in the heart and the skeletal muscle, as we demonstrate here. And when we look further into the heart, we were actually intrigued to see that the expression of the marker gene of this reporter is not heterogeneous at the beginning across the entire ventricle, and it's actually absent from the atria. But very quickly, it'll populate the entire ventricle without any sort of specific space. So we're interested to see what is the wave of maturation in this critical period of time. So overall, this story tells us that there is a genetic program embedded in the gene regulation to drive the RBFOX1 specifically in the postnatal period. And the RBFOX1 mediated alternative splicing actually have a significant contribution to the cellular maturation process of cardiomyocytes. Combined with our earlier observation in the P38 MAP kinase pathway in the postnatal sort of chamber-specific modulation, it's interesting that both RBFOX1 and P38 are significantly implicated in the disease as well. They can contribute to the disease progression, and they can actually be used to targetedly ameliorate the disease progression. On the other hand, they both are intricately important for this developmental period between the neonatal hearts transitioning to adult hearts. And so I think this the lesson we learned from this two line of studies is that the neonatal heart maturation turned out indeed shed important lights into the pathogenesis mechanism in the adult hearts. Like vice versa, I think the neonatal hearts actually, perhaps from a sort of a philosophical sense, already employed many of the mechanisms that we discover in the adult hearts to shape and to support the process during the neonate maturation progression. I wanted to conclude by thank the funders for our study in USA. I've been funded continuously by the NIH and Department of Defense, as well as American Heart. And I also wanted to thank the new grants that we received from the Ministry of Education and the NMRC from Singapore, and several other private foundations. And with that, I wanted to share with you the recruitment opportunity at the Duke NUS CVMD department. And we're looking for associate and assistant professor on the tenure track. I welcome you to join us. And if you have opportunity, please visit us in Singapore. With that, thank you very much.